Just a minute. No, not this one. Yeah, this one. Okay. See, already use the word extracellular, isn't it? When you take a cell, so inside the cell is intracellular, the whole outside environment is extracellular. Here the diagram I have shown in some other different angle. So here when you observe the diagram, you're having this, the whole blue one, the blue color picture at the center of the slide is part of cell. In the cell, this is the cytoplasm part. And this purple color one, which I am shading on the diagram, is the plasma membrane. Okay. The simple nature of plasma membrane I showed, it didn't show any phospholipid bilayer, something like that. Because here we are discussing about the major role of plasma membrane in movement. What is the major role of plasma membrane in movement of molecules? Some different processes are there. Okay, that's why I took a different idea regarding plasma membrane. So here this curved one is plasma membrane. Understand that point first. Now inside the plasma membrane, definitely you will have cytoplasm and different cell organs. So this blue part, this whole blue part is the intracellular part. Then obviously outside plasma membrane would be extracellular part. Okay, so this is extracellular part outside the cell, and this is plasma membrane, and this is intracellular part inside the cell having cytosol or cytoplasm. Both are same. In some textbooks they'll give cytosol instead of cytoplasm. That's all. Now here we are saying three different processes: phagocytosis, pinocytosis, receptor-mediated endocytosis. So these are different processes which are involved in movement of major molecules through the plasma membrane from extracellular matter into the cell. How they are moving. First I'll take the example of phagocytosis. Phagocytosis. Cytosis always stands for taking into cytoplasm. Taking into cytoplasm. Wherever you find the cytosis word, that stands this meaning. So what is taking in? The phagosomes will be taken. What are phagosomes? See, I'm underlining on the slide. Phagosomes are nothing but bacteria or bacterial products. Okay. So when there is any infection, in other words, we can say, if there is any infection by bacteria, how that infection happens? Your body is taking those bacteria or bacterial products in the form of phagosome. Now when coming to the what does mean by phagosome, just see the picture. The plasma membrane forming a pseudopodial-like structures. Just shading on them, see. Okay, the pseudopodial-like structures which you'll find in amoeba. Like that the plasma membrane is folding. And it is taking the yellow particles. See, the yellow particles are nothing but the bacteria here. So the yellow particles will be taken in because of the pseudopodial-like evacuation of plasma membrane, and it will be ingested. Ingest means what? Taking it. Ingested in the form of a vesicle, which is called phagosome. So the whole process is called phagocytosis. So in short, phagocytosis is a process where the bacteria or the bacterial products will be taken in by the cell. Okay, that is meant by phagocytosis. Suppose if the same plasma membrane is taking in liquid particles, liquid droplets from outside into the cell. Here they have shown the droplets and this color. So these liquid droplets will be taken in again by same pseudopodia like evagination of the plasma membrane, just shading on the screen. See. Okay. So they will form vesicle-like structure. Vesicle is nothing but bubbles with pinocytic. What does mean by pinocytes? Taking in liquid material. Okay. So this is one process. So here from the outside of the cell, if bacteria or bacteria particles are taken in, that is called phagocytosis process. If liquid particles are taken in, that is called pinocytosis process. And sometimes you will find endocytosis. What does mean by endocytosis? I took an example and explained. See, this is plasma membrane. This is extracellular material. And this is cell. Okay. So in the cell, you are having the plasma membrane structure. Now what happens on the plasma membrane structure? If you take the lipid bilayer will be having some receptors. Receptors which receive only some molecules which they recognize. Here they have shown receptors in blue shaded one, and only some molecules among all are taken in. This yellow bacteria was not taken in. These droplets like things are also not taken in. Only the red color particles are taken in. This is because these will be recognized by the receptors of the plasma membrane. So only they will be taken in, and that process we call as endocytosis. And specifically receptor mediated endocytosis. Here I have taken an example, clathrin. Clathrin is nothing but a protein. Okay. So protein which will be taken in because of the receptor mediated vesicles. So these are the three different processes by which various molecules will be taken up by the plasma membrane. Have you got any doubt? Phagocytosis, just repeating the whole summary. Phagocytosis, pinocytosis, receptor mediated endocytosis. Okay. When we take phagocytosis, this is the process by which solid particles, for example bacteria or the virus infection or the bacterial products infection, this will be done through the plasma membrane by forming phagosome. Remember the word phagosome, that carries importance. Okay, that's why the process is called phagocytosis. Nothing but eating up solid material. That's all. In simple, to remember, phagocytosis is eating up solid material by the plasma membrane. Okay. Now when you take pinocytosis, in the same way, we can simplify taking in liquid material. Kind of cell drinking. In some books they'll give like that also, crude words. But good word. Pinocytosis is nothing but cell drinking. Through the plasma membrane, liquid material will be moving in. Okay. But biologically we are talking it as pinocytic vesicle or pinocytosis. Then endocytosis. Endocytosis always happens only when there is receptor recognition. Receptor will be there on the plasma membrane which will recognize what molecules it has to take in. That is called receptor mediated endocytosis. So these are the different processes by which molecules will move through plasma membrane into the cell. Am I clear about three processes? If you have doubt, let me know. Bhumika, no doubts?
No doubt, ma'am.
No doubts, Anna? Okay, fine. Bhumika, yourself?
Ma'am, can you please that pinocytotic
vesicle. Yeah, sure. See, when you take this liquid droplets, just making them a bit red shaded particles. Okay, so these are the liquid droplets. Okay, now yellow ones are the bacteria, and the red ones are the receptor mediated particles. So this will be excluded, this will be excluded, this will be excluded, this will be excluded. Only the red shaded liquid molecules only we are talking, because we are talking about pinocytosis. What happens? The plasma membrane will form pseudopodial-like structures. Okay, false. Okay, pseudopod-like invaginations, the plasma membrane form like this, and these particles will move into the plasma membrane like this, and it will be taken in, forming a bubble-like structure called vesicle. Here it is taking the liquid droplets, no? That's why we are saying it as pinocytotic. The cytotic doesn't matter any point over here. Like understanding water molecules are getting it. So making it complicated, they used the word pinocytotic. Okay. Only the liquid molecules will be taken in by the plasma membrane and they form vesicles. Vesicles are nothing but the bubble-like structures. The bubble-like structures with water molecules which came in, or the liquid molecules which came in. That forms a pinocytotic vesicle, which will be formed in pinocytosis process. Okay? Okay, Bhumika?
Yes, ma'am.
Then, if this is clear, I'm going to share few questions related to the osmosis and diffusion. Try to